I'm Dr. Alexandra Benchoua. I'm a research director at IStem, and I'm leading the team Research and Technological Innovation, which is also in charge of the stemCARE platform. So the stemCARE platform has a total of 6 facilities. The first one is a biocell facility.
The cell biology platform meets the needs of the Institute research teams for all methodological aspects of human pluripotent stem cell culture and their derivatives. Our mission is to ensure the quality control of these cells and, if necessary, the optimization of their protocol. In particular, we are in charge of cell karyotyping for the Institute-owned teams, as well as for external academic or industrial teams, in collaboration or as a paid service. Our platform is equipped with a Metasystem automated microscopy workstation for chromosome identification and analysis. We are able to detect anomalies larger than 5 megabase.
The second is genome editing facilities.
Here at the genome editing facility at IStem, we generate cellular models for pathological modeling and screening approaches. So we do that by genetically modifying human pluripotent stem cell lines using the CRISPR-Cas9 technology. As the reaction is not complete, we use robotic devices to isolate cellular clones, which are checked for correct editing. Each clone is then subjected to classical quality controls before to be transferred to the research team which has requested the cells.
The third facility is the bioproduction facilities.
So you are now in the bioproduction platform. The activity of our team is to produce huge amount of cells dedicated to all the teams and also for partnership. So here we are using 2 main technologies. The first one is the compact select. So the robot is inside the hood and can make all the steps of the amplification of the cell. And we are producing more research-grade bank of cells. Then we have another machine.
So this is a Climax Prodigy, and it's mostly used for the proof of concept for the clinical cell culture level. So here you can connect the medium on the top. It's directly connecting on wires. You have a peristaltic pump to distribute the medium here on external vessel for cell culture. Or you can choose also to work directly inside the Prodigy. So you have an internal chamber for the cell culture. And after, you can put directly your external vessel in the warm environment inside the incubator.
So at the end of the process, we have a huge quantity of cells that need to be freezed, and it's a critical step. So this is a fillet system, which is a machine that can manage a box of 96 vials at the same time. So this is a step just take less than 2 minutes, and several boxes can be freezed for 1 batch of cells.
So once the cells are produced, then they can be safely stored in our liquid nitrogen tank in the biobank. Then we have 2 facilities dedicated to the molecular and phenotypical characterization of the cells. So the first one is the NGS facilities.
So here we are on the next generation sequencing platform, and we use this platform mainly to study the gene expression, so at the tissue or sample level, such as bulk RNA-seq, or at the single cell level with the single cell RNA-seq. This technology is used first for the disease modeling to try to identify a specific signature of pathological cells, or for the cell therapy to study the heterogeneity or homogeneity of the cells after a specific process. So first, the cells are barcoded using the Chromium system, and then the library are fully automated with the Hamilton star, which is a liquid handler. And then we'll use the sequencer to sequence the sample. After this, the data are fully analyzed on the platform, and here we can visualize the different cell types and go in deeper in the analysis for the researcher team.
The ultimate goal of IStem is to find treatment for patients.
Our screening platform is composed of all the devices allow us to develop a cell-based assay using the Bravo bench shell and test 1,000 molecules using our new Beckman platform. On this new platform, we have an echo which can dispense nanoliter, whereas the Biomake i7 can dispense microliters to milliliters. In this new platform, we can now realize screening not only in single dose, but also in serial deletion and soon in combination.
Once we have produced the cells, the point is to study them.
So we are now on the cell imaging platform on which we can analyze the plates with the cell culture. We have the possibility to make different kind of analyses on live or fixed cells. High content screening machines allows us to analyze a high amount of cell culture conditions, and at the end we can obtain quantitative analysis for the whole plate and the different conditions. This allows us to have a high amount of quantitative data at the end of the analysis. We are now on the dark room where we have the different equipment for the conventional microscopy, from very basic epifluorescence or bright field microscopes to the high resolution confocal we have here. We make a lot of three-dimensional imaging, and we also have, for example, a spinning disc on which we can make some different kind of functional imaging.
We decided to create the stemCARE platform to bring very high-level and innovative technologies to people at IStem, but also to all the scientific community in France and abroad.